Welcome everybody this evening. It's a real pleasure to see you here. For those who don't know who I am, I'm Professor Pauline Walsh and I'm the Pro-Vice Chancellor and Executive Dean for the Faculty of Medicine and Health Sciences. It's a long mouthful, isn't it? And so I'm chairing this evening's session, and I'm really delighted to introduce to you Professor Paul Cool. And these types of inaugurals are really special because they enable us to hear about the work that colleagues are doing. And for many of us, we won't know that work. We won't necessarily work closely with somebody. So for both myself and the Vice-Chancellor who's with us this evening, this is really a pleasure that we sit and listen to colleagues talking about their work. I'm going to say a little bit about Paul, but I know that he's going to sort of orientate us to his career during his talk. But he qualified in 1988 in Rotterdam and joined Birmingham for his surgical rotation. And then he came to Oswestry in Shropshire for his orthopaedic rotation. Anyone who's not been to Oswestry, to the hospital out there, it's a lovely setting, really nice environment to be in. He was awarded the prestigious European Travel Fellowship from the British Orthopaedic Association, and looks like he has visited Florence, Vienna, Leiden and Stockholm. So in 20,000, I always say that wrong, he was appointed as a consultant orthopaedic surgeon at Robert Jones and Agnes Hunt Trust. He's got an interest in foot and ankle and orthopaedic oncology. He set up the bone tumour unit in Oswestry, which clearly is something very important and impressive. And he's the lead cancer clinician during 2000 and 2023 for Robert Jones and Agnes Hunt. So he's helped Oswestry, in a leading capacity to become a national specialist in accredited bone tumour treatment centre. So there's only 5 in the country, so again, a big accolade. And he's been the lead fundraising for a new bone tumour treatment centre and theatre complex at about 15 million at the hospital. He's also director of laboratories there, and he's the lead UCAS accreditation person. So he's been many things. So he's past president of the British Orthopaedic Oncology Society. He led the Biobank for Health and Disease to promote genomic sequencing in orthopaedic conditions, Meridian Award to promote genomic medicine in 2018, and he's been a statistical advisor for the European Journal of Hand Surgery. His special interests are anatomy, diagnostics, data collection and outcomes, machine learning and statistics. So you can clearly see how he transcends different professions and specialties. So with that, I'm absolutely delighted to invite Professor Paul Cool to give his inaugural lecture on orthopaedic objectivity.
Well, thanks, Pauline. A very kind introduction. It's very kind, and thank you all for coming as well. Very much appreciated. Also, thank you for people online, and of course for Keele University. I seem to have managed to persuade the appointments committee to give me this job, so thank you very much. I was going to talk about orthopaedic objectivity and really about the adventures of me over the last 20 years or so, or actually really before that, but to outline my journey in a way, from starting with my interest, as Pauline just said, in diagnostics, radiology, pathology, but also anatomy and mechanics. And to combine this, the best thing to do, I think, is orthopedics, in my view. And I like doing that with surgical treatments, and I'll give you some illustrations of that, and I hope you enjoy it. you see, this is on the wrong side. Funny thing. Starts off here. I was a bit of a chubby baby. I'm going the wrong way. A bit of a chubby baby, as you can see here, sitting on my grandfather's lap. I did get a bit thinner, but I really put it on here to show you that I had some hair in the past, you know. Yeah, a haircut was necessary to go to the medical school, which I did. I went to medical school in Rotterdam. It's not that far, and if you go with sustainability nowadays, you go with a bike from Kiel to Rotterdam. It's about a day of cycling, and then about 6-7 hours on the boat, and then you can go with the underground in Rotterdam if you want. There's this yellow line you can see there, so the Hoek of Holland, and then if you see there, sort of in the middle, Koolhaven, which is one of the metro stops in an area called Kool in Rotterdam. And if you're there, you see this is Koolhaven in Rotterdam, and you see there at the right the Euromast, and right at the back you see the Rhine when it enters in the North Sea. What you can also see on the picture is the medical school, the big white building, and the university hospital, which is the grey building in front there. So that's where I did my medical training. I had to do a little bit of duty in the national service. Did manage to get out very quickly. I'm very pleased to say, and you can find me. I'm just over there. Nobody really was very keen to be there. All conscripts, apart from the lady in the middle. She was actually a professional vet studying, because these are all doctors and pharmacists in the class. Anyway, ending up 1988 in Good Hope Hospital. Sorry that this picture doesn't look quite so nice, but it is quite hard to make Good Hope Hospital look nice, even here in the blossom in the spring. But I did have a great time there, to be honest. And this was my first patient, and I remember this well to today. A 40-year-old guy who used to play rugby when he was younger, and he then obviously became not obviously, but he became a trainer. And he thought, well, one more time I want to play rugby, one more go. And he played rugby and he broke his neck. And you can see that here for the medical people under us. You can see the fracture there of his vertebrae, and off in the traction he goes. And my boss just said, oh, well, just take him to Oswestry, which is the spinal center for it. I've never heard of Oswestry at that time. Just take him over there in the ambulance. 1.5 hour trip, if you're lucky, in the ambulance. And so, you know, in this traction, I was saying, well, what would I do if this traction comes off? And the boss said, well, just put it back on. I don't know how to do this. But anyway, so I held these calipers on this guy's head in the traction, in the ambulance journey. And it took quite a while, quite a bit longer than 1.5 hours, because I asked the ambulance to drive very slowly, because I really didn't want these things to come off. And so then I ended up in Oswestry. You can see that here, an aerial photograph of the hospital. It's really in a countryside hospital. And it is one of the few hospitals in the country which has this coat of arms. You can see here on the bottom, you see Dame Agnes and Sir Robert Jones's crest in there, and then the Shropshire leopard in the middle in the red. Now, it may come as a bit of a surprise to medical people here, but orthopedic surgeons are actually medically qualified. That wasn't so the case for many years, and in fact, people who started orthopedics, you see on the right there, you see Hugh Owen Thomas. He was actually one of the first medically qualified orthopedic surgeons. His father was just a bone setter, and he never had any orthopedic or medical training as such, but he did realize that this was important, so he sent his son, Hugh Owen Thomas, to study medicine before becoming an orthopedic surgeon. And people may know him from the Thomas splint, which is the one he described. And then his nephew is on the right, his nephew. he became very famous as a brigadier in the army, and he set up the hospital together with Dame Agnes. So one most important orthopedic operation is probably total hip replacement. And you can see here quite ghastly hips, to be honest with you. I mean, there is joint space narrowing, as you can see here. There's some cysts, as you can see, osteophytes, and some sclerosis. And initially, the idea was that, well, why don't you just take the hip out and put one of these door knocker things in here? As you can see here, you can see this is called a hemiarthroplasty, which is half a hip, if you like. And it wasn't until the 60s that Charlie realized that just putting in just the head itself is not good enough. You have to put in the socket as well, and that's when he designed this total hip replacement. And this is the best orthopedic operation we have at the moment, and it's going to be hard to beat, I think. Charlie, he did this, but he never did a randomized controlled trial. People are always full of these randomized controlled trials, so you have to do them, and I can see that, but you wouldn't want to do this on this. Now, this lady is laughing her head off on this randomized controlled trial she's in. It may sort her hip, but I must say I prefer this, and I really don't think you need to randomize controlled trial to see if the parachute works. And you may not sort your hip pain though. These hip replacements don't always go well, and we had Percy the Peacock roaming around the hospital for a while, and then he died. And you can see here a tribute to Percy on the front of the hospital, made all of hip replacements, which are all welded together. So in 1988, I joined the surgical rotation, as Pauline just said. I did some research in Birmingham, studying limb salvage in children, and then orthopedic training in Oswald Street till about 2000. And then I managed to convince them to appoint me when I finished my training. What next? And it says here, this in Alice from, I thought it was quite funny, where to go? Well, it depends where you want to go. No, I don't know. The first thing came across, and this was really out of necessity, was this child who had quite severe bone pain. And this is a very small tumor called an osteoid osteoma. It's entirely benign, but it's nevertheless very, very painful. And at 2000, the standard treatment for this was really to excise it. And you can imagine you take a block of bone out and the tumor is in there, then it will cure the pain. It's a bit crude, I think. I always felt that. And if you look here, this is a pathology specimen. You can see this red cherry, and you can see that that's what this tumor is. It also sees that you take quite a lot of bone out for taking out a small little tumor, which is entirely benign. And so, well, is that really necessary? Also, the problems is it's really quite small, 2 centimeter max, difficult to find. Some anatomical locations is even harder than here. You can imagine there are fracture quite easily. Then you may have to put a plate on it to reinforce it, and it may get infected, so you can see all the problems. And see, this boy, which I just showed you the X-rays of, you see here, this is a CT scan. And they tried to in another hospital, before they sent it to us, they tried to take this osteoid osteoma out, and you can see here the healing response from the little hole they made in the bone. You can probably just about see here the, where they went in to try to get this lesion. But that lesion is just still there, and no surprise, the pain is still there as well. So this is not good for a little 6-year-old like that. So what we devised together with the radiologist, I think it's been really quite remarkable, is in the CT scanner, put a needle in that lesion and then just heat it up. And if you heat it up for 5 minutes at 90 degrees, it just inactivates the lesion and it just completely goes. So this is him post-op. This was his previous surgical scar from had done previously, and now he only has a little plaster for this. So it's been a remarkable treatment, I think. Great progress for children, but also for adolescents. It's quite interesting when you look at this mechanically, and you want to have a little hole, you drill a little hole, you want to then put a tube over it. It won't fit because the drill makes a hole which is smaller than the tube is, so you need to use an eccentric drill, which they also use in the oil platforms, I believe, to do that. So you use this eccentric drill to get down, you can put a sleeve over it. Now you can see here on this, it's very difficult to see on the x-rays, but you can see it easy on the scans, this lesion. If you would have to excise this, that would be completely wrecking an adolescent's knee if you would do that. You would need a knee replacement for that. So to burn that with one of those probes has been revolutionary, I think. It's interesting, this tip is completely cold. It's not some hot poker you put in. It is actually a radio frequency sort of device. Interestingly, the diameter is it can treat maximum is 1 centimeter. And it's completely independent on the energy you put in. You can put more energy in it doesn't matter. You cannot get a bigger radius of cell death or necrosis after that. So if you have a lesion which is more than 2 centimeters in diameter, you have to put in more probes, and I've done that with livers, but never done in orthopedics. And then together with the radiologist and Badash Lalem and Victor Pellicino, we devised this. We used this umbrella. You can maybe just about see it there. These umbrellas which come out, but it's quite tricky to deploy in bone. But that's the only way you can treat bigger lesions like that. And again, that's been, I think, quite revolutionary. So I say in Alice in Wonderland, we have to maybe believe in possible things. But I think 30 years ago, this was felt to be impossible, and this needed to be done with big operations. I think another thing there's been real progress in, I think, is in metastatic bone disease or spreads of cancer in the bone. Bone cancers usually come from either thyroid, breast, kidney, lung, myeloma, prostate. They're sort of the common culprits. We often see breast and prostate particularly, although prostate a bit less more recently, but breast and kidney are the common ones. And you see here, where do they go? They go to the spine, the humerus, the shoulder, and the hip. And particularly the hip is more important, I think, because if you break your hip because of a deposit of the cancer there, then you can't walk, you'd be bedbound. And in orthopedic surgeons, and most people not orthopedic surgeons say, oh, I just pin the hip, this is easy. And you can see this here. This lady had a breast cancer in a bone, and you can see that here. And then somebody put a nail in it. Now you can probably also see that the fracture is not really reduced. It looks a bit sort of like an amateur would not be that proud of it if you were a DIYer, I don't think. But also, more importantly, of course, the fracture is not going to heal because it broke in the first place because of a cancer in there. And the inevitable happens that you got here later, and this screw, you can see it at the top, this pokes in the hip, and there's only going to be more painful. And they've got a variety of patients coming through the clinic like that. This is another one. There's a bigger nail. And you think, well, it's less likely to break. Well, if the fracture doesn't heal, then it will break. And you can see there at the top, the nail is definitely broken. And those 2 screws at the bottom they put in to stabilize it, they're also broken. And to take those out, it's really quite tricky. As you can imagine, with these broken screws with the locks in at the bottom, this whole nail in the femur bone, to try to take that out is really quite hard, as you can see here. So I thought, there must be a better way. And I think the internal fixation, if you think about it, these nails or these plates or whatever you put on, they are made for fractures which are following an injury, we call traumatic fractures, and they are really designed for the fracture to heal. And if the fracture doesn't heal, that device, just like a paperclip, if you move it often enough, it will break. And generally speaking, if it's more than a year, then the chance that the nail doesn't hold it is going to be quite strong. So if a patient has a survival of more than a year, then maybe this is not the right thing to do. Like I said, pathological fractures, on the other hand, which are fractures which are caused by cancer, they will never heal. Movement is painful, and if the fracture doesn't heal when you live long enough, then this nail will fail. Also, which I think particularly troublesome, is that if you have a cancer in the hip there, and you stick a big nail through it, then you spread all these cancer cells right down through the whole thing. And it can't be better. You're just making it worse, I think. So I don't think never thought this was a very good idea. You can see here an example of a patient who's got a left hip. We always do it from the left side, that's the way you look at it. So this is the left hip. So this is a left hip breast cancer. And what are you doing with this? And then orthopedics, so we need to pin it. Well, no, the first thing you need to do is, well, what are the options? Options are do nothing, do some sort of operation, give some sort of drugs, maybe some radiation, or maybe a combination of all of the above. But from surgical point of view, the most important thing is this going to break? You know, if it's not going to break, you may just best not leave it alone and maybe give some radiotherapy, you know? So how can you predict if it's going to break? Now, they have got this guy called Merrill on the basis of about 30 patients, not a big study to be honest with you, designed this score. And if you score, you can score from between 4 and 12 based on 4 things, and you can have a score from 1 to 3. Now, people will probably agree this is in the hip, so that's for this patient not a problem, so you get 3 points for that. I don't think most people will disagree that this is a mixed lesion, so it's some bone destruction and bone formation, so that's 2 points, you get 5. And then the pain and the size of the lesion, actually, it's very, very subjective. How do you know how much pain somebody is? How can you put this in a number? It's very, very hard. And the size of the lesion is not really described very well in the paper. Is this a third of the lining of the bone? Is it a third of the whole bone? It's actually not clear at all in the paper. And consequently, the variation you have between scorers is enormous. And if you see here, this is between different orthopedic surgeons who scored a series of 100 x-rays, and they're easily 2 point either way out. You know, so that's a 4-point difference on a 12-point scale. I think it's quite a lot, you know. But not only that, the same person actually, 3 months later, is also 1 point either way wrong. You know, so this is not a good way of predicting if something is going to break. And I think this is a little bit, well, obviously very subjective. But I think it's a little bit like this white square you see, which has a value of 255 pixel value, and then if this grey square, which is 130. I set this at 130. If you now say this has to be black or white, what would you choose? It's neither black nor white. You have to choose one. What would you take? Well, actually, I'd probably say black, to be honest with you, but it's 130, which is above 127, so it should be white. You know, but I think there's very few people who would say that is white. You know, so that, I think, is inherently the problem. Now, what we've done, and this is Emma Howard, who did her master's here in Kiel, and she done phenomenally well, measuring how much weight people could put on their leg. So if they could put full weight on it, then obviously the chance that you break your leg is almost nil. And so we worked out with this rock curve. It's a bit fancy, but basically if you score, if you can put 85% or more of your weight on your leg, it's not going to break. And this is, the rock curve will show you better than the Merrill score, but it's also more reproducible. You can see that. And in a way, what you do, you're measuring pain. You know, if somebody has no pain, then they can put their full weight on it. And if they've got a lot of pain, they can't put their weight on it. So in a way, I think this is a better measure of pain. Still not perfect, but it's definitely better. And so this lady, going back to her, she had she could put only 72% of her weight on her leg. So, yeah, probably needs something doing. And that, you know, you can see the orthopedic surgeon thinking, can we do an operation now? We really need to do an operation. But they say, well, actually, hold fire still, because I think these operations are really big operations, and they're not without its risks. And if the survival is 3 months or less, or you think that somebody has a survival of less than 3 months, whether you do anybody any favor is a big operation which takes at least 2 months to get over. Perhaps not. So perhaps you need to explore non-operative options. And if you think the survival is up to a year, then I think some of these nails they've put in may well be a suitable solution. However, if you live longer than a year, or expect to live longer than a year, then that nail will almost certainly fail, which is what the literature shows as well. So we really need to have a look and see, can we predict that? Now, we've done some work on that as well, and this is a little bit more complicated, but we call this the quadruple A assessor for breast cancer. This only works for breast cancer, but you basically look at 3 blood tests, the albumin, the adjusted calcium, and the alkaline phosphatase, which are quite commonly markers used in the blood and the patient's age, because interestingly, if you're older with breast cancer, then it's usually less aggressive than if you're young. Young people with breast cancer usually have a more aggressive type of breast cancer. So if you fill all these results in, and you can say here is 5.73, that's well above 1, so we would expect this lady to survive for well over a year. And therefore, she gets the door knocker, yeah? Which, proud of a nice operation you've done, but it worked actually very well. And just give you an idea, another one, this is another hip we've done, and this lady actually already fractured, because you can possibly see that a little bit of the lesser trochanter here is already fractured off. And she had a similar door knocker, maybe, if I'm critical, a little bit oversized here, but it was the smallest one I had. Function really, really good. You know, Karen will probably say I'm a little bit pervy. It's a lovely young lady with breast cancer. She had liver cancer actually, but she wanted to do this pole dancing she really loved, and she couldn't do that afterwards, you know, and I don't think she would have done that with any of the other treatments. Problem with the nails, not the nails, problem with these, massive implants is they can get infected, and it's a real disaster. And you can see here it's quite obvious that this plate, which is on this patient's knee, is infected. But it's not always so obvious, and sometimes you can look at it clinically, sometimes you have a variety of blood tests we use, sometimes we do some pathology, but that's not really specific. That won't tell us what bug it is. We can do microbiological cultures, whereby we take a sample and then we send it to the lab, and then the laboratory will try to culture this bug and then tell us what it is. But it takes 2 days or so, and you know, you can get some false positives, you can get some false negatives. It's actually not as easy as you may think. So the idea was, can we use genomic sequencing to find the bacterial DNA? If you look at Moore's law for the development of computers, you can see that's that dotted line, that the genomic developments have been much, much quicker and faster than even Moore's law. Back in 2001, it was $1 billion to sequence the first genome, and now it's way lower than £1,000. You know, so it's enormous. Revolution again, I think, in medicine, this genomic evaluation. Just to give you the sheer scale of this, it's just enormous. You have 3 billion base pairs. So these are pairs, so there's 6 billion nucleotides you have, an average person has in its body. So if you would want to work out the length, obviously a very long, thin string, but that would be 20 billion kilometers. You know, it's a massive long stem. So how long is that? Well, that is about 133 astronomical units, you know, so 133 times to the sun and back, or 18.5 light years. 18.5 light hours is the distance of the light to travel that distance. This is a phenomenal amount of information which is stored in every human. Now recently, this is one of the main advances in this genomic sequencing, to have this nanopore sequencing. And what that is you have a graphene monolayer where you put in these nanopores, which are proteins which allow the DNA to go through. And if you then put a potential across the membrane, then you pull that DNA through those pores. And when it goes through the pores, the voltage across the membrane slightly changes depending on the nucleotide it represents. So you can then read real time, as you go along, you can read the DNA code. And there are only 4 nucleotides, so you've got one A, C, T, and G, and that will come out in sequence. So now, when you want to know how many salmons there are in the river, you're certainly not going to count your salmons. What you do, you take a water sample, you just find out how much salmon DNA is in the water. And also, you can find all the other fish as well. This is quite impressive, and this is what a lot of environmental people do now. So the idea to use this to identify infections, I don't think is that way off. It's quite cheap. We've got this little machine here, as you can see. The error rate is a little bit higher than traditional genomic sequencing, but nevertheless, it's quite acceptable. It's also quite easy to do, says he, who's never done it himself. But here, this is in the International Space Station where they done that. They have, you can see there the little device there. I've cheated a little bit, to be honest with you, because all the DNA extraction they did on Earth, and then that makes it running through the flow cell a little bit easier. So what we do, we take some blood or tissue or fluid and we extract it. And then the big thing is, that's what they don't do in the space station, is you need to centrifuge it to get the DNA out. And that's 8,000 G. So that's not a small centrifuge. And I think there, that would be quite dangerous doing that in the space station, I think. So then the purified DNA goes into that cell, and in real time you can get it on your computer, and then you can analyze it with known sequences, because there's whole big databases which say that sequence of that bug, that sequence of that bug. So you can then look it up in the database. So one of the first cases I've done is a patient with a bacterial infection, with 2 or 3 bugs in. 2 hours of sequencing and 620 megabyte of file you get just with AGCT, you know, 620 megabyte of it, you know, it's a big text file. And it looks a bit like this, you know, you've got an identifier and then you've got just the code, and then the bit underneath, don't worry, is just some computer language for the quality of the code. But the code itself is just a bit in the middle, these big TTs, GTAs and so on. So if you then see what sort of DNA is in there, then you won't be surprised that 19,500 of these reads were human. It was quite good because we took it off human person, so that's quite reassuring. What you can also see is that a lot of these other ones in green are all primates, and that's also perhaps not so surprising, because lots of the genes we have are just in common with primates like chimpanzees and gorillas and orangutans. You see here, I quite like the human in the bottom there. I think it perhaps looks a bit like an orthopedic surgeon. But if you now sequence this as a bacterial database, so if you now say the same sequences, we look at the bacterial database, then the bugs which were in there, which we grew and cultured, you see they're all there. So that gave me quite a good motivation to carry on with this. Well, this must be a good thing to do. And Holly, you've seen, and she's in the audience today, and you see here when she's not surfing, she is sequencing, and she's doing a PhD on this to see if we can improve these methods. She's very busy, and you know, this is part of the things she's doing to improve the DNA extraction, the read lengths and the sequences, reduce time, and etc. Would even be nicer if we could do this even from the blood beforehand, and we can work on that as well. What we did find in that is that you always find some bacterial DNA in any sample. You can, of course, there's bacteria everywhere around us, so you can have some contamination. So I think it always has to be interpreted with a background of, is this really an infection or not? And one of the things you can do is you can count the neutrophils, and if you have more than 5 in a high-powered pathology field like this, then if you have more than 5, then it's infected. Won't tell you what it's infected with, but it will tell you it's infected. Now, this special stain makes these neutrophils grow red. Now, you can see there's more than 5, but I asked you to count them, then you probably give up before you started. I would anyway, so this is tedious. Humans definitely want to do that. It's a computer job, isn't it? And then like here, from how many neutrophils are in this picture, you know, which is one of the slides of our patients. And if we've done some artificial intelligence learning on it to try to get the computer to see these neutrophils, and these, in theory, these algorithms are quite straightforward. You know, it's a bit like what's 1 and 1 and 1 and 1 and 1 and 1 and 1. I don't know, lost count. Well, you can't do addition then, said the Red Queen to Alice. And you can see that rather tedious to go through all this, but computers are really good at doing these tedious things. And there we go, on 4 neutrophils, which it found. And they said, well, are these the real ones? Is of course the question. Now they've done the, so the ground truth is here. So if you go back, this is what the computer found, and this is what the real truth is. So it misses 4, which annoyed me, but what it did do is all the ones which it identified were all correct. So in other words, it's quite specific, but the sensitivity is still a bit low. But I'm working on that, and hopefully with some help from Theo, I'll get there. And I looked at these images, and actually those neutrophils do look a little bit different. Another problem with it is that if you use different pictures, why would you want to count neutrophils on guys coming out of the bus? No, I just tried it on this picture, but somebody's eye comes up as being a neutrophil, and obviously one of the things illustrating that computers are not very good in generalizing. So they'll just count this as a neutrophil, and it's a little bit like looking at the wheel, and you see the Ferrari sign, and so, well, this must be a Ferrari, but obviously it's a crappy Golf, as you can see, you know. So, yeah, and I think you always have that, and computers will always be fooled by that, and I think it will always need to have some sort of human interaction, if you like. Some other concerns I have with computing methods, like I wanted to share with you, and one of the things I've sort of asked is ChatGPT. People are all on about it, how wonderful this is, how fantastic. So I asked, well, can you give me 5 highly cited papers written solely by Paul Kuhl? The first one, 1961, I wasn't born yet. This is the identifier, you know, the DOI, of this validity of opinion change produced by deviant communicants. Interesting, because the actual, if you look at the DOI and look it up, it was obviously not written by me. It's to a completely different author, a completely different title, and the only thing which is the same is the DOI. And well, this, you know, this just makes it up, and the computer just tells you what it thinks you want to hear, you know, but it clearly is complete and utter nonsense. So imagine if you have 2 doors here, and you need to say, well, one is a computer and the other one is human. How would you do that? How would you ask that? And of course, a very simple thing you could do is say, well, what's pi? And humans are bigger 3.14, and then probably some people may get a bit further, but oh, here it is, 200 decimal points. You know, no problem, the computer will spit it out. Not a single human, I think, will ever do that. So one way of finding out who's behind the door would be to ask, give me pi 200 decimal points. But the more interesting question, perhaps, and I think you could probably read it, if Apple made the car, would it have windows, you know? And the thing is, I thought it was quite funny, but you know, the computer just doesn't find this funny at all, and it says yeah, well, it's proprietary software, Apple would probably use their own software, and so this is obviously not understanding the question, and I think that's what computers are. You know, so computers are really good in doing calculations. They're also really good in memorizing things. They store information incredibly well. But as I showed you, they don't really generalize very well. And also, I think, quite wasteful if you say, I want to be waking up tomorrow at 8 o'clock in the morning, and every time the loop will go round and round and round, checking every second. It's got a huge amount of energy. We wouldn't do that. So before you go to bed, you will ever think about it. You know, certainly wouldn't think about it now. Humans are awful at calculating things, I think, in general. Memory, you may think we've got a good memory, but I think it's hugely biased, unreliable, and also changes over time. And sort of, I'm myself guilty of sometimes making the story a little bit more exciting, shall we say. But humans are incredibly good at high cognitive task, problem solving, reasoning, understanding, and particularly young people and children. They learn fantastically well, and I think we should maybe perhaps use them a bit more in our thinking, because they usually have a lot of intuition, children. So yeah, I think we brainwashed them by training them too quickly, perhaps, but it's a personal thing. I think this objectivity is, I think it's really hard. It's really hard to be objective. I think the computers can help with that, but certainly on their own, as I hopefully demonstrated with a few examples, that they're not the solution on their own, and they will need some humans to supervise them. I think it's some sort of human interpretation is necessary and essential. So some sort of symbiosis in a way. Well, in that, ladies and gentlemen, I think it's important that you have some fears you have, you try to overcome. I myself, I'm absolutely scared of heights. And you see here, this is the new church in Delft. It's the, I think the highest but one church tower in Holland. And thought I'll climb up that. I'm really scared of heights, to be honest. And when I was up there, so this lady said, "You look really scared." That's because I am really scared, you know. But I did manage to take this picture of Delft, as you can see there, and the area down really flat, as you can see in Holland, where I grew up. And it's not too much shaking, I think, so I did manage, but I still didn't like it. And when we were there last summer, I declined to go up again, so still it wimped out. I'm okay though on flat grounds in here, or when it's on the ground and it's not like that. It's still high. Snowdon is higher than the church, but I'm okay doing it like that. And you can see here, my son Oliver wanted to go up Snowdon with the bike, which is what we did. And well, the Hawkeye, and you can probably see, I thought I'll be really clever. I'll go with my e-bike. That would be no problem. I'll just zoof up that mountain, no problem. Not realizing that most of the time you cannot cycle, not even with the e-bikes. You have to hold that whole heavy battery with you as well. So it wasn't quite such a smart idea, I'm afraid. So yeah, it's probably better to taking a lighter bike, to be honest with you. But we did have a great time. It took about 5 hours, I think, to get up, and half an hour to come down. It was great to come down. Time, I think. This is my 3D printed sundial, which is digital, so you can see the shadow on the table there at 11:30, I think. Quite clever, I think, these clever design. Anyway, thanks to the Orthopaedic Institute, which have been funding some of my studies in striker implants, who have also done that. Lots of thanks. I'm not going to name them all, but Karen, I mean, honestly, how she can put up with me, I really don't know, but thanks very much. Surgeons, Mr. Potter, Richard sadly can't be here. Rob, Matt, and Barin have been fantastic colleagues, and the three Davids, my colleagues in the foot team. Radiologists, like particularly Naomi, Dencia, Victor and Radesh with the burning of the tissue have been fantastic. Our pathologist, ADN, I work very closely with, as well as a colleague, Fernanda, Roberto, and Daniel. Nurses have been fantastic support over the year, and also the new secretaries have now been very good in welcoming me. Research laboratory, lots of them are here. Karina, Charlotte, Helen and Jade, and students keep me sane, I must say. Management have also been quite helpful in supporting my academic role, so thank you to Ruth and Dawn. And colleagues in Kiel here, particularly Christian, he's been absolutely fantastic, supportive, I think. Theo and Charlene. Don't forget patients. I've got 2 more slides to show you about patients. This is, I think, the best present a patient can give you. She had spina bifida, and she had the reason it's on the right is because her left leg had an amputation for an uncontrollable tremor. And she was so fed up with this and said, "Can't you just take my leg off?" No, of course, and take your leg off. So he took her leg off, and she was so delighted with it that she wrote this on her, tattooed this on her leg. I said, well, you can't get a better compliment than that. It's just amazing. And Adrian here, all these patients are given permission, by the way, of showing their slides. Adrian here had one of these infected implants, and he had his his leg taken off at the hip, and here he is 5 years later, coming down the mountain, a mountain bike, an e-bike, of course, as well, with one clip-on leg, so he just goes with his one leg down the hills, and you can see, I think he looks a bit scared, but he's also having great fun, which is what it's all about. Ladies and gentlemen, thank you very much.